Assalamu alaikum. This is Dr. Muhammad Bilal from Bilal Hijama Clinic, Pakistan, and today we are going to discuss the anatomy and physiology related to blood because this in this series we are discussing those topics which are very important to perform hijama correctly. So you know that through hijama we extract the toxin containing blood. So we should also know about the normal blood or the fresh blood which is circulating in our body, and what are different parts of this cardiovascular system which are making up this system, and what are the components and how this whole system is working. So today we will discuss briefly about the cardiovascular system. So the circulatory system, it includes composed of three parts. The first part is fluid, which is blood. The second part is the system of blood vessels through which this blood is it travels throughout our body. And the third part is the heart, which is the pump which is providing the force for this blood to circulate throughout the body. So one by one we will discuss all of these three parts. So the first part, which is fluid or blood, it is actually composed of again two parts. First is the liquid part and the other part is the solid part. The liquid part is termed as plasma, and this plasma is it contains water, and it contains certain proteins, enzymes, clotting factors, all of the parameters which are normally tested in our biochemistry tests, such as the cholesterol, uric acid, and SGPT, SGOT. All of the enzymes, they are present in plasma. And if we minus the clotting factors from plasma, then the remaining part will be termed as serum. So the difference between serum and plasma is that the clotting factors are present in plasma, while serum does not have the clotting factors. So after that, the remaining part is a solid part, or the cellular part of the blood, which is composed of three types of blood cells. The first are the red blood cells or the erythrocytes. The second one is the white blood cells or the leukocytes, and the third type is the platelets or the thrombocytes. So these are the three cellular types of blood which are present in the solid part of the blood. The liquid part was plasma. The solid part composed of three types of blood cells. Now we will discuss the red blood cells or the RBCs. They are red in color because they contain iron, and where iron is located inside the RBCs. RBCs are actually when they are mature and when they are released in the blood into the bloodstream, their shape is donut-like shape, and they are flattened from the middle, and their outer edges are swollen like a donut. So the donut is empty from the center, but they are not empty; they are packed, and the mature cells are of donut shape. So RBCs make up about 99% of the total blood cells. They are abundant, so that is why the color of blood is red. It appears red to us because RBCs are in majority. Now RBCs contain a functional unit which is termed as hemoglobin. Hemoglobin is actually a compound of heme and globin. One heme and four globin molecules make a single hemoglobin molecule, and a single hemoglobin molecule can carry up to 4 oxygen atoms. So the basic function of hemoglobin is to transport oxygen throughout the body. Whenever we are inhaling, whenever we are breathing, the oxygen is getting attached to the RBCs which are present in our lungs, and the carbon dioxide is released outside when we are exhaling. So the main function of RBCs is to supply the oxygen to all parts of the body, to all cells of the body. At every cellular level, this oxygen reaches with the help of the RBCs. So when the RBC is carrying oxygen, the color of the RBC turns bright red. So and if the blood which is coming back through the veins, and the oxygen has been consumed, and this blood is actually bringing back the carbon dioxide which was produced as a byproduct of oxygen. When cells consume oxygen, they release carbon dioxide, and this carbon dioxide is again collected by the hemoglobin. It attaches and then it is termed as carboxyhemoglobin, and then it turns a little bit blue in color, and then this carbon dioxide containing blood is transported back through the veins to the heart, from where it is sent to the lungs to get purified, to get rid of that carbon dioxide and to attach the fresh oxygen inhaled by the lungs. And then through the vein, this fresh blood is again transported back to the heart, and then heart pumps it throughout the body. Now, we have discussed a little bit about the circulatory system. Inshallah, we will discuss it in detail later. But right now, we are focusing on RBCs. So, RBCs are in majority, and as I have told you that the main function of RBC is to supply oxygen to all cells of the body. Now, the things which are necessary to manufacture RBCs are iron, first of all, then copper. Copper makes its absorption from the GIT easy, then cobalt, nickel, vitamin C, riboflavin, nicotinic acid, pyridoxine. These are the different nutrients which are required to build or to produce the RBCs. Now, the process of blood formation or the process of RBC formation is known as erythropoiesis. Erythropoiesis or blood formation, this process takes place inside the bone marrow, and when a child is born, the all bone marrow of the body is actually red in color because of the rapid production of RBCs throughout the bone marrow, because the body is growing, and at that time RBCs are required in great quantity. So that is why all of the bone marrow is red at that time. But after puberty, the bone marrow of the long bones, the bone marrow of the ends of the long bones remains red in color, while the bone marrow which is present between the shaft of the bone turns pale yellow in color, and it turns to fat basically. So that's where the blood formation takes place, and a very important hormone which is known as erythropoietin is responsible for the production of RBCs, and this erythropoietin is produced in the kidneys, and there are some sensors present in the kidneys which can sense the saturation of oxygen in the blood which is passing through the kidneys. Whenever this oxygen saturation falls below a certain threshold, the neutrophil is the erythropoietin is started to secrete from the kidneys, and then it stimulates the bone marrow to produce more cells and more rapidly. So, and when this process starts, bone marrow is stimulated by the erythropoietin, it starts producing RBCs in great number, and when the required number of RBCs has reached, what happens that oxygen saturation of blood increases, and when this blood passes through the kidneys, the sensors sense that now the oxygen saturation is sufficient, so it means that no more blood formation is required, so they stop the production of erythropoietin, and then in this way in the bone marrow is cut off, the production of RBCs is stopped, and a certain level of RBCs is maintained in the blood. So that is how erythropoietin is very important in the formation of red blood cells. And that is the reason that in certain sports, you might have seen that some athletes are tested positive in a dope test, and actually the drug which they test in that test is actually erythropoietin. Because if an athlete uses erythropoietin, his RBCs will increase, and oxygen carrying capacity will increase, so his stamina will increase, then he will be able to outperform the other athletes, and that is cheating. So that is why erythropoietin is a banned drug whenever it is used before a sports event. So this was something related to the RBCs and about the erythropoietin. Now we will move to the second part, which is anemia. So if the RBC production is below the normal level, then anemia may result, and anemia means low hemoglobin level. And there are different types of anemia. Sometimes it is due to iron deficiency, sometimes it is due to vitamin B12 deficiency, sometimes it is due to some genetic defect such as thalassemia, and sometimes it is sickle cell anemia, which is also a genetic defect. So there are different types of anemias, and during anemia we try to avoid doing hijama, if especially the hemoglobin level is below 7. So we try to avoid hijama at that time. And another important factor is hematocrit, which is packed cell volume measured in percentage. It increases in the case of patients of polycythemia. It increases above 50, otherwise it should be under 45. So if it increases, then it might be suspected that the patient is suffering from polycythemia. At that time, hemoglobin will also be at above normal, and it decreases, the hematocrit decreases in anemias and in cirrhosis of liver, in pregnancy, and in some other conditions. The RBC lifespan is about 120 days, and after 120 days, what happens that these RBCs, which are very minute in their size, but when they pass through spleen, there are certain capillaries which are very thin like a hair, and RBCs pass through these capillaries one by one. They cannot pass side by side. Even two RBCs cannot pass through the splenic blood capillaries. So what happens that even the single RBC has to squeeze through the capillaries, and when the lifespan of RBC has reached to 120 days, then their cell membrane gets fragile, and when they try to squeeze through the spleen, they get ruptured. And when they get ruptured, what happens, the hemoglobin is broken down into its components, heme and globin. Then globin is further divided into bilirubin and biliverdin, and then this bilirubin is stored in the form of bile. It is transported to the liver, where it is synthesized as bile, and then it is transported to the gallbladder, from where it is again excreted in the large intestine. And the heme or iron is stored, which is used for the production of new RBCs, so it gets recycled. So the heme never leaves our body. That is why the patients who are suffering from thalassemia major, and they have to go through blood transfusion in 2 or 3 weeks, or sometimes on weekly basis. So the iron which is coming inside their body rapidly, it does not leave the body, and then iron overloading results, which is a very dangerous condition, and that can damage different organs if that iron is deposited on certain vital organs. So chelation therapy is mandatory at the time, and I would like to tell you that hijama is also a very effective procedure to remove that excess iron from the body. So we have also performed hijama on thalassemia major patients, and we have seen great results. A patient who was suffering from thalassemia major, and his serum iron level was 17,500. It reduced to under 5,000 after 6 to 7 sessions. So hijama is also a very effective way of treating the thalassemia major patients for the target of removal of excessive iron, which is a very big problem for them. Okay, so we have discussed the RBCs, their life cycle, their erythropoietin, which is responsible for the bone marrow stimulation and the production of RBCs. Another factor is ESR, or the erythrocyte sedimentation rate, and this is the rate at which the erythrocytes settle down. It increases during pregnancy and menstruation, and it increases in tuberculosis and in any kind of inflammation. It also increases in autoimmune disorders such as the rheumatoid arthritis, and it decreases in allergic conditions or sickle cell anemia, polycythemia, and severe leukocytosis. So these are, I think, that enough information about the RBCs, or the red blood cells, which are also termed as erythrocytes. The next we will discuss the white blood cells, which are the defense line of our body. And the white blood cells are of two types. First is granulocytes, the other is agranulocytes. They are produced inside the bone marrow, and they are pluripotent stem cells, or the hematopoietic stem cells are present in the bone marrow. And in lymphatic cells, there are also lymphoid stem cells. So the lymphoid stem cells produce B cells and T cells, the lymphocytes, and the bone marrow produces the myeloid stem cells, and these myeloid stem cells produce the basophil, eosinophil, erythrocytes, platelets, monocytes, and neutrophils. So all of these types of white blood cells are categorized into two types. The first is granulocytes, which contains grains-like structures in them, and they are three types. Three types of white blood cells are granulocytes, which are neutrophils, eosinophils, and basophils. And the agranulocytes are two types, monocytes and lymphocytes. So these are the total 5 types of white blood cells. Neutrophils mostly destroy the microorganisms by lytic enzymes, and they are mostly bactericidal. So they kill the bacteria, and this is their if their number is increased in the CBC report, then you should think about the bacterial infection, that the patient is might be suffering from bacterial infection, because sometimes a false positive result also appears when the patient is on steroids. So if a patient is taking steroids, then you should ignore that increased number of neutrophils in that patient. Eosinophils are actually the defense mechanism against the parasites such as the Taenia solium, or the other types of worms, other abdominal worms which are present inside the body. So eosinophils are increased due to the presence of parasites inside the gut, mostly gut. And then the basophils are the white blood type of blood cells which have a very important role in healing processes, or in allergy or acute hypersensitivity reactions, and they release heparin, histamine, and slow reacting substances of anaphylaxis, bradykinin, and serotonin. So whenever basophils are increased in the CBC, then you should also check the serum IgE level of that patient, because that patient might be suffering from allergy. Then the monocytes are the precursors of the tissue macrophages, and the tissue macrophages are the white cells which can engulf the patrolling bacteria or the pathogenic germs inside the tissues, and they engulf them, and through phagocytosis and through the lytic enzymes they can kill that bacteria. The lymphocytes which are produced by the lymphoid stem cells are of 3 types: cytotoxic T cells, the helper T cells, and the B cells. The cytotoxic T cells kill virus infected and damaged cells. So lymphocytes are basically against the viruses. So if the patient is suffering from a viral infection such as dengue or hepatitis, then might be the lymphocytes are increased in his CBC. Then the helper T cells help the cytotoxic T cells and B cells in their immune functions. The B cells produce antibodies, so whenever infection occurs, the B cells are activated, and they are actually the memory cells also. They remember that this infection was caused by which specific pathogen or bacteria, and then they start producing antibodies against that. And whenever that infection tries to invade the body again, and the next time, the antibodies are already prepared for that, and they, through the lock and key mechanism, they destroy that microorganism before causing any problem. So the lymphocytes are of 2 types, T lymphocytes and B lymphocytes, and both of them are stored in lymphoid tissue of lymph nodes, spleen, bone marrow, and GIT. And the B cells provide humoral or blood cell mediated immunity. The T cell provides cell mediated immunity. The helper T cells are destroyed in AIDS, and the cytotoxic T cells attack the pathogens and also kill the body own cells if not suppressed by the suppressor T cells. In autoimmune diseases, that is the case. The T cells are decreased or destroyed, so they cannot stop the cytotoxic T cells. They cannot stop them, and the cytotoxic T cells keep destroying our own body's cells, and that's how the autoimmune disorders are triggered. And then there are immunoglobulins, there are different types of immunoglobulins. For example, I told you about IgE. IgE is very important because it is a very good indicator of presence of allergy in the body, and most of the IgG and IgM, they are now obsolete. They are not very much accurate about the as a test. They do not tell us exactly about which infection is disturbing the body. So, and then the pus which is formed after the infection is actually the dead bodies of the WBCs and the bacteria or the foreign bodies, and the cellular debris which has been the collateral damage. So that is how the pus is formed. There are different lines of defense. For example, macrophages, neutrophil invasion, the second macrophage invasion, then increased production of granulocytes and monocytes. These are our lines of defense. So this was about the defense system which is managed by the white blood cells. Then we come to the last cellular part of the blood, which is platelets. And basically platelets are responsible for stopping the loss of blood which is caused by due to some damage to the vein or to the blood vessel. They plug that hole in the wall, and they basically trigger the clotting factors cascade, which results in the formation of fibrin, and then fibrin makes a meshwork or network in which platelets are trapped, and then the RBCs are also trapped, and they plug the hole, and they also contract. The fibrin contracts, and it heals the cut in the blood vessel. And when the blood vessel is repaired and the cells of the wall are multiplied and they have replaced the damaged ones, then the anticoagulants are activated and they remove the clot, or they dilute the fibrin back into the liquid form, which is the clotting factors. And the lifespan of platelets is 10 days. So these platelets are also very important. Their normal values are from 150,000 to 450,000 in our CBC report. So if the patient is suffering from thrombocytopenia, then it means that if the patient's platelets level is below 1 lakh, then you should not perform hijama. So I'm telling you about in reference to hijama, because we are actually discussing the anatomy and physiology, which is very important for performing hijama correctly. So we will relate to hijama time to time, and if the patient is suffering from dengue, you should avoid hijama. If the patient is suffering from cancer and he is going through the chemotherapy, then it also decreases the platelet count. So you should be very much vigilant in those patients who are receiving the chemotherapy, and you should not perform hijama before performing their CBC to observe the platelet count, because the blood will not stop if the platelets are too low in number. Okay, so this was all about the cellular part of the blood. Now we have completed the fluid portion. Inshallah, in the next video we will cover the remaining parts, that is the blood vessels and heart. Inshallah, we will discuss those in details. Keep tuned, and inshallah we will cover the whole topics as soon as possible. Thank you very much. Assalamu alaikum.